Hello, my name is Joshua Mugabi. I welcome you to Topic 1, Cell Biology. Our learning outcome is D. Analyze the structures of plant, parenchyma, collenchyma, sclerenchyma, xylem and phloem, and animal, epithelial, cardiac, areolar, fibrous and skeletal tissues to assess their roles in physiological processes, disease diagnosis, and level of organization. Our learning activity A. In groups, learners observe and discuss micrographs, images, prepared slides, or prepared slides of plant tissues, epithelial tissues, and connective tissues. Relate the structure and location of tissues to their function. Then make a write-up with labeled drawings and present it to the class. Observation and discussion of plant and animal tissues. Item 1. We are going to observation of plant tissues. Students are provided with micrographs and prepared slides of parenchyma, collenchyma, sclerenchyma. They are asked to observe and identify the tissues. Task: identify the 3 plant tissues observed. State 2 observable structural features of each tissue. Name 1 location where each tissue is found. Identify the 3 plant tissues observed. They are the parenchyma, collenchyma, and sclerenchyma. State 2 observable structural features of each tissue. Parenchyma. They have thin wall cell walls. They have large intercellular spaces. Collenchyma. They have unevenly thickened cell walls at corners, leaving elongated cells. Sclerenchyma. They are thick lignified walls. They contain dead cells with narrow remains. Name 1 location where each tissue is found. Parenchyma are found in the cortex and pith. Collenchyma found beneath epidermis of stems. Sclerenchyma found in the vesicular bundles and seed coats. Observation and discussion of plants and animal tissues. Item 2. We are looking at the observation of animal tissues. Students examine prepared slides of epithelial tissues, connective tissue. Task: identify the tissues observed. State two structural features of each tissue. Give one example of a specific type under each category. Identify the tissues observed. Epithelial tissues and connective tissues. State two structural features of each tissue. Epithelial tissues. They are closely packed cells. There is little or no intercellular spaces. Connective tissues. Cells widely spaced. Presence of intercellular matrix. Give one example of a specific type under each category. Epithelial tissue example is squamous epithelium. Connective tissue examples are blood, cartilage, and bones. Let's go to relating structure and location of tissues to function. Item 3. We are looking at structure-function relationship in plant tissues. A learner asks why collenchyma and scalenchyma both provide support but are found in different parts of the plant. Task: relate the structure of collenchyma to its function. Relate the structure of scalenchyma to its function. Explain why their locations differ. Relate the structure of collenchyma to its function. Collenchyma has unevenly thickened walls. Cells are living and flexible. They provide support to growing parts without restricting growth. Relate the structure of scalenchyma to its function. Scalenchyma cells have thick lignified cell walls. The cells are dead and rigid. They provide strong mechanical support and protection. Explain why their locations differ. Collenchyma occur in young stems and leaf stalks. Scalenchyma occur in mature tissues where rigidity is required. relating structure and location of tissue to function. Item 4. We are looking at the structure and function relationship in animal tissues. Student, observe squamous epithelium from the lining of the alveoli. Task: describe the structure of squamous epithelium. State its location. Explain how its structure suits its function. Describe the structure of squamous epithelium. It is thin, flat cells arranged in a single layer. State its location. It is located in the lining of the alveoli in lungs and capillary walls. Explain how its structure suits its function. Thin cells allow rapid diffusion of gases. Short diffusion distance increases efficiency. Relating structure and location of tissues to function. Item 5. We are looking at connective tissue adaptations. A prepared slide shows blood tissue. Task: identify the tissue. State 2 structural features. Relate these features to its function. Identify the tissue. It is blood, which is a connective tissue. State 2 structural features. Cells suspended in plasma. Presence of red and white blood cells. Relate these features to its function. Plasma transport nutrients and hormones. Red blood cells transport oxygen. White blood cells provide immunity. Marking emphasis. Correctly identify tissues. Describe observable features. State precise locations. Relate structure clearly to function. Use correct biological terminologies. Conclusion. The structure and location of tissues are directly related to their specific functions in plants and animals. Thank you for participating in Topic 1, cell biology, our learning outcome D. Analyze the structure of plant, parenchyma, collenchyma, sclerenchyma, xylem and phloem, and animal, epithelial, cardiac, areolar, fibrous, and skeletal tissue to assess their roles in physiological processes, disease diagnosis, and level of organism. Our learning activity has been A. In groups, learners observe and discuss micrographs, images, prepared slides, or prepared slides of plant tissues, epithelial tissues, and connective tissues. They relate the structure and location of the tissues to their functions. They make a write-up with labeled drawings and present it to the class. If it has been useful to you, visit me at Mugabi Joshua Biology on YouTube.